Well, I'm still in Portland.
I know. I
Um,
know.
but thank you for having me. I'm a little intimidated to be in front of a bunch of plant nerds because I definitely know more about mushrooms than I do about plants, but this is one area that I've got a pretty good grip on. So I'm looking forward to telling y'all what I have learned. Um, I guess I would like to clarify that I am not a professional, so there's plenty of things that I don't know, but this is a subject that I'm pretty passionate about, and so I have read many papers and discussed this with a lot of people. So hopefully I know enough to tell y'all what I've learned. Um, and yeah, so that's a bit about me. And because I don't come from an academic science background, my aim is really to make this kind of knowledge accessible for other people who are like me, who just may be passionate and interested. So I'm going to try and use common names as much as possible and speak in a very basic language that hopefully everyone can understand. That said, if there's something that you didn't get, feel free to ask questions in the chat. I'm going to keep a chat window open, or just if you have a question about anything. Okay, so a little bit about what got me interested in mycoheterotrophic plants is that 9-ish years ago, I went to my first Mycological Society meetings, so I'm really interested in mushrooms. And I didn't know anything about mushrooms really at that time. I knew how to identify chanterelles and that was it. But our speaker that month was Steve Trudell, and he had a presentation about the mushroom genus Tricholoma. And I didn't know what that was. But he said that the edible mushroom matsutake had an association with this plant. This would be a good time to show you the plant. Okay, so hopefully y'all can see my screen there. Moment. So this plant, there we go. This plant is the candy stick. And I didn't really know what this mushroom was, but I had never seen this plant before, and I thought it was incredible. It looks like a candy cane. Um, it was just like one of the coolest plants I had ever seen. And so I really wanted to find that thing. So just a moment, I'm going to try and get my chat window open here. Yep, there we are. Okay. So it was going to be a couple more years before I found it, but I did find some other mycoheterotrophic plants. So let's look at what these things look like. So this one is called coral root. It's an orchid. You may have seen these. These are the really young shoots that come up, and at this point it's really hard to tell exactly which one it is. This one is called pine drops, and it's pretty recognizable at this stage with all the flowers drooping down on it, but then oftentimes you'll see it later on looking like this, and those stalks will persist for, at least a year afterwards. You get these dry woody stalks. There's this one, which is the ghost pipe. This is one of the most common ones that I see in my area, and it also grows throughout North America and elsewhere. This one is called pine sap. I like the common name shy shrimp on this. No one else calls it that yet. But my thought is that it looks like a little shrimp that's kind of like nodding its head and a little bit afraid to come up. So especially when they're young and they have these nodding heads like this, I like to call them shy shrimp. Please feel free to pass that on, but most people do know this one as pine sap. And this one is called gnome plant. I see this one especially near the coast. Okay, so when people talk about these things, they sometimes call them saprophytes. So I'd like to break down that word really quick. Sapro means rot or decay, and phyte means plant. So that would mean that they are plants that are decaying and breaking down dead material in order to get nutrition. However, that's not quite correct. We'll talk about why. But just wanted to clarify that even though we do know at this point that they're not saprophytes, even some fairly recent texts, this book from 1994 calls some of these things saprophytes, even though we know that they're not. And for whatever reason, people have been very slow to try and get away from the idea that they were doing this, or at least calling them that. And if you just Google, is Monotropa a saprophyte? So Monotropa is the ghost pipe plant. And the first link that came up 3 years ago anyway was this one that confirms that yes, it's a saprophyte, but that plant is actually incorrect. Now this is the third link that comes up if you type that in, so we're slightly improving, but it's still in there. Okay, so I made this meme because this is something I'm pretty passionate about, and they are definitely not saprophytes. So why did people think that they were saprophytes? Probably because they don't have any green parts on them, and they look kind of like mushrooms, and mushrooms do actually break down dead stuff and eat them, or fungi do. And so there was just this association that was kind of made with them. And since, plants photosynthesize and they have chlorophyll that helps them do this, and that's why they're green, ones that were not green kind of makes sense that they would just do something else like that, but that's not what they do. And so why are these recent sources saying this? If we've known for a while that that was probably not true. So in 1881, a scientist, Kamiensky, observed that the roots of this plant were completely enshrouded in the mycelium of fungi, so that cobwebby stuff underground that's the actual body of the fungus. And so he was like, it seems like the fungus is definitely playing some role, because otherwise it would be blocking the plant from getting nutrition. So it must be doing something. And even in 1909, people knew that that fungi were required for the germination of orchids. Some of the mycoheterotrophic plants are orchids. We'll talk more about that later. But for whatever reason, they couldn't get the idea that they would still be giving nutrition to the fungi later on. So let's talk about what they are doing. Here we go. Oh, really quickly. This is the root of pine drops, and you can see all of this furry stuff that's on it. That is mycelium of fungi. So this is kind of a modern take on what that scientist was seeing when he was saying, hey, the roots are completely enshrouded in fungi, so it must be doing something here, or else it would be blocking it from getting nutrition. Okay, so the word mycoheterotroph, myco means fungus or mushroom, hetero meaning different or other, and troph meaning gathering nutrition from. So it is something that is getting nutrition from a source that is outside of itself. And it's a special kind of mycorrhizal relationship. So for folks who are not familiar already with mycorrhizal relationships, myco again means fungus or mushroom, and rhiz means root. And there are a couple of different kinds of mycorrhizal associations, but what it is fungi that have associations with the roots of living plants, and they do nutrient exchange. Okay, so we've got this underground bit where we can see that they're connected to each other. And the plant is doing photosynthesis, and it's producing all of this carbon, these sugars, from that photosynthesis process, making its own food with the sun. And then it gives a lot of those sugars down to the fungus, which is attached to it. And then the fungus attached to the roots, either penetrating the roots or on the outside of the roots, depending on what kind of mycorrhizae it is. And what that does is the fungus body, all of this cobwebby stuff, helps to extend the root network of those trees or other plants. So it really helps them to get water and also phosphorus and nitrogen and other nutrients out of the soil that those plants aren't very good at getting on their own because their roots are relatively coarse. So fungi that have mycorrhizal relationships are dependent upon these plants for that relationship, and the plants are dependent upon those fungi for that. So they both need each other to survive. Now, not all fungi do this, but mycorrhizal fungi do. Okay, so here's where the carbon's going. And then we have our mycoheterotrophic plant, and it gets in on the action. And we've got the roots of the mycoheterotrophic plant here, and that attaches to the fungus. And as far as where our nutrients are going, they're coming down from the plant into the fungus, and then they are going into the mycoheterotrophic plant, all attached underground. So it's very difficult to actually observe this happening, which is why they're so mysterious for such a long time and why people probably thought that they were saprophytes. And as far as the word saprophyte, no plants do that. So that word is moot altogether. Yeah. Okay, so this is what they're doing. That wasn't known for a very long time, even though it was observed that those plants are completely surrounded in fungus. Nobody could for sure say what the fungus was doing and how it was working. So in 1960, Eric Bjorkman did this experiment to try and figure that out. So he had pine sap plants, and he knew that there was fungus on the outside of them. There's also fungus that is attached to the roots of these trees, and you can see with a microscope that each of these things have fungus on them, and so that's how he knew that. And what he did was he took a piece of sheet metal around those plants like this, and he buried it over 2 feet, maybe 3 feet, to block those roots of the plant off. So they would still be covered in the fungus, and the fungus would still be on the roots of trees as well. And if it was just the fungus that was doing something for the plant, then there should be no problem, right? The plant should still be able to live and do its thing. But that's not what happened. He came back the next year to check on the experiment. And here are the ones on the left that were in the original portion. And then a year later, he comes to check on them, and they are kind of looking like failures at life. So this indicated that there was actually a connection there between that fungus and the tree, giving some sort of nutrition through that network. And then he actually went further on that, and he used radio-labeled carbon. So this is a special form of carbon that is not usually in the environment. So radioactive carbon. He drilled holes in the nearby tree and then poured that radioactive carbon into the holes of the tree. And the idea was that he wanted to try and see where that radioactive carbon was going to show up in the places nearby. Would it be in the other plants? Would it end up in the mycoheterotrophic plant? And he checked back on it 5 days later. And he found that it was indeed in the mycoheterotrophic plant, in the pine sap, and to no great extent was it in those other green plants. So that means it wasn't just like leaching out into the soil. It seems like it was attached to this fungus and directly going into the mycoheterotrophic plant. So this was a big discovery. And this also helped for people to understand how mycorrhizal relationships just in general were working. And then he also went further than that, and he cultivated the fungus that was found on the roots, and he cultivated it in the lab, and then he added extracts of the pine sap, that mycoheterotrophic plant, to the fungal cultures, and it had a growth stimulating effect on that fungus. We're still working out exactly how that works, but somehow it stimulates the fungus to grow. And I just want to take a moment to acknowledge that he accomplished quite a lot without many of the technological advances that we have now, such as DNA. A lot of this stuff is easier to figure out now by just, running the DNA, and we'll talk more about that later. But it's pretty cool that he was able to do this experiment and figure out where that carbon was going. Okay, so there were still many questions after that, such as, are the mycoheterotrophs giving anything in return? We're still working on that one, but I'll talk more about it later. Which species of fungi are involved? When he was doing this experiment, he was speculating about which fungi there might be. There was some sort of boletus that was nearby, or a bolete. And so he thought that maybe it was the boletus, but it turns out that it wasn't. A lot of times the fungi are not actually fruiting at the same time that the mycoheterotrophic plants are up. So you can't just assume that because there's a mushroom nearby that it has anything to do with it. And usually you see these mycoheterotrophic plants in the summer, and the fungi can often be in the fall. So how did the relationship evolve, and how common are these types of relationships? So as far as how common they are, it has evolved around 40 times independently in several different plant lineages. So it's not just all in one area. It's within the Ericaceae, the Orchidaceae, even one liverwort is known to be mycoheterotrophic. And then there is also partially mycoheterotrophic plants in other families, and some stages of ferns and club mosses. But the most commonly studied groups are in the Monotropoideae, which is in the Ericaceae, and also the orchids. And we're going to mostly focus on that for the rest of the presentation. And as far as how they evolved, it seems like all the closest relatives are photosynthetic plants. So they probably sometime in their lineage were photosynthesizing and then somehow moved away from that. Okay, and no matter how they got to be there are some commonalities that more or less fit in all of them. They have very reduced vegetative parts, so the leaves are almost non-existent or very small. They don't have very many stems. It's just a very reduced sort of plant. They live mostly underground most of the year, with underground forms being more like storage areas than forms for absorption. So with a few exceptions, they mostly have root systems that are kind of coarse, ball-like or coral-like things that don't have a lot of little rootlets or anything like that. And they generally have an absence of stomata, the holes for gas exchange that aid in photosynthesis, because they're not photosynthesizing, so they don't need those. They often live in very low light forest floor conditions, so places that it wouldn't be really handy for a plant to grow otherwise, because they're not getting access to the sun in order to make food. And generally they have fungal host specificity, at least among the mycoheterotrophic plants that are fully mycoheterotrophic. Some have some green parts and are not fully mycoheterotrophic, so they do some of their own food production. They have great reductions in the size of the seed, so they have very tiny seeds, and a lack of differentiation of the embryo of the seed. So basically they just don't have much of a food store within that seed, so they are going to need some external source in order for the seed to germinate. And they also have a very large number of light, tiny seeds for wind dispersal. So here's some examples of what these things look like. Here you can see this is pine drops. These are really reduced leaves, obviously no green parts. There's just one stalk. It's just here for the business, and it's just making flowers, and that's it. And it's very hard to find pictures of some of this stuff online, so I did have to do a little bit of digging up some mycoheterotrophic plants. I wouldn't recommend doing that in general because you can potentially kill them, but I did it once for science. And here are the roots of the ghost pipe. And you can see that there's kind of a like cottony stuff on the outside of that, and there's fungus all around it. But this is basically it, you know, it's just in this one spot here. They're not branching out into a lot of areas because it's depending on fungus to bring food to it. And here's the coral root, kind of a similar deal, really coarse coral-like roots. And I see there's a question. They do generate pollen. Yes, they do generate pollen, and they have insect pollinators as well. I know that the ghost pipe has been observed just with bees, I think, and no other pollinators yet. Okay, and then here is an example of those really light seeds without the differentiated embryo, so very small. Okay, and as far as the seed germination, in 2004, there was a study that had to do with pine sap seed germination. And what they did is they buried the seeds of the pine sap in a place where it was already known to grow. So they knew that the conditions were right for it. And they had these mesh nets that they would periodically come and check on and see what was happening with the seed. So in stage 2 is where the seed has actually germinated, and none of the seeds made it to stage 2 and germination unless they were colonized by this fungus. And it seemed like the presence of that fungus being attached to it is what made the seed germinate. They were able to take the fungus from those seeds and do some DNA work on it. And then they also took fungus from the trees that were nearby, the willows, and they did DNA on that as well. And they found that it was the same fungus, tricholoma cingulatum. As far as I know, that fungus doesn't have a common name, but it's a big gilled mushroom. And in other areas, this same mycoheterotrophic plant associates with different species of fungi that are within that same genus. But what happens is that it's still pretty specialized within a given area. So it might vary by geography, by region, by elevation, but within any specific small area, it's very specific on one host, and that is true in general of the mycoheterotrophic plants. And then there is Cryptophallus, which is the only known liverwort that is mycoheterotrophic. It's also the only known non-vascular plant that is mycoheterotrophic. And it looks like this. Pretty weird. Obviously no green parts, doesn't even have roots on it really. So what they did is they found that there was all of this fungus that was just hanging out on the bottom portion here. And very similar to the last one, they grew this fungus in the lab. So they knew that the fungus did form a mycorrhizal relationship with these particular birch seedlings. So they were able to figure out that the fungus that was associated was a Tulesnella. Usually that's like a crusty kind of thing. It doesn't really make real mushrooms. And then they also were able to find that it was mycorrhizal with the birch. And this one was kind of a twofer because they figured out what the fungal host was for this liverwort, and they also figured out that in this case, this fungus was mycorrhizal. And up until this point, they had only thought that this fungus was saprotrophic, meaning that it fills the ecological role of breaking down stuff that's already dead, rather than being in this mutual association with trees. Okay, So among the mycoheterotrophic plants, there are fully mycoheterotrophic plants, and then there are ones that are partially mycoheterotrophic. And a big group of partial mycoheterotrophs are the orchids. All orchids in the wild are mycoheterotrophic at seed germination. So their seeds will not germinate without the presence of fungi and generally very specific fungi. Within growing settings, y'all maybe know this already. Those conditions can actually be replicated just by giving it the kind of nutrients that it needs in order to germinate. But at least in the wild, they always need a fungus for that. And within orchid mycorrhizae, it's not really like the same sort of association that's happening within the Ericaceae plants. There are little fungal coils that actually go inside of the cells of the plants, and then they kind of fall off inside, and then it ends up eating the fungus. So it's really kind of a weird thing where it's, you know, it's eating it instead of just the nutrients going back and forth. And as far as orchids that are photosynthetic at some point, they generally pair with saprotrophic fungi, and that is different than other fully mycoheterotrophic plants. So that's weird, and we'll talk more about that in a bit. And a few orchids are mycoheterotrophic for their entire lives, so they never photosynthesize, or they at least take stuff from fungi for their entire lives. Outside of the tropics, the fully mycoheterotrophic orchids pair with mycorrhizal fungi. So that's a very long-term stable carbon source because it's attached to trees that are long-lived. We'll talk more about that in a bit. Okay, so since this mycorrhizal partnership would really be a better long-term strategy because the fungi are paired with trees, which are a really good long-term carbon source, why on earth would orchids partner with saprotrophic fungi, which just kind of exhaust their food source and then die? So these are some ideas, and nobody totally knows for sure why, but probably it has to do with orchids, many of these orchids being able to photosynthesize later in life, so it's not really necessary for them to have a reliable carbon source beyond that seed germination phase. They can take care of it themselves later. Okay, I hear y'all have some audio interference. Okay, let me know if you miss anything specific in the chat and I'll try and go over it again. Most orchids, the highest diversity of orchids are happening in the tropics. So it could have to do something with habitat. There is a lot of soil erosion in the tropics. So there's a lot of really fast nutrient cycling as there's all of these rains that happen all the time and wash the soil away. So it may be that the soil isn't as nutritious, so it doesn't provide as much of a good source for the mycorrhizal fungi to have a good source of food. And it may be that the extended growing season in these warm, wet places is just a better place for saprotrophic fungi in general. But these are just some ideas. Nobody necessarily knows exactly why they're more prone to associate with saprotrophic fungi than mycorrhizal fungi. However, outside of the tropics, fully mycoheterotrophic orchids do pair with mycorrhizal fungi. So it may be that there's something specific about that environment that's causing it to happen. And here is an example of how successful that can be. This is Erythrorchis altissima, the world's largest mycoheterotroph, and it can get stems that are up to 32 feet high. it does grow kind of upward as a viney thing on the stems of other plants. So that gives it a little more height. Okay. And this one is kind of an exception in that it turns out that it associates with quite a few different fungi, not just one. And they found in this experiment that it was associated with at least 37 species, and these are mostly wood decay fungi. And the way they figure this out is they can measure the types of carbon and nitrogen, the different isotopes of them in the fungi and the plant. And so they're able to actually determine that not just are these fungi present associated with it, but they're actually contributing to the nutrition of the plant. Okay, and here's an example of what some of those fungi that were associated look like. You may be familiar with this one if you're a mushroom nerd, but if you're not, that's okay. Trichaptum, it's a very, very common mushroom. Flavia, this is another really common one. We see it all winter long up here. And then here's another one that associates only with saprotrophic fungi, but this one only has a single saprotrophic fungus host. And that host is Satherella candoliana. So if you've got any mushroom nerds out there, you might know that this is a very common fungus and it happens to occur all over the world and can have just really big fruitings. So basically the takeaway is probably that it's able to associate with just this one saprotrophic fungus because it is so common and widespread. So it might represent a pretty good partner, even though it will eat up its food source and then go away. Okay, so outside of the tropics, though, most mycoheterotrophs, they actually outside of the tropics, all mycoheterotrophic, fully mycoheterotrophic plants do associate with mycorrhizal fungi. And the benefit to that is that it allows for a longer term stable source of carbon because it's attached again to this tree that's very long lived. And so it can give that carbon from the tree for, potentially hundreds of years. And the great part about this for the mycoheterotroph, other than getting free food, is that the tree requires that association with that fungus. So there's no way that it's going to make defenses against that fungus because it needs the fungus to survive. So it actually, it's a really good way to tap into that because there's no way that the tree is going to make defenses against it because it needs that buddy. Generally speaking, there is host specialization. And that is typical of parasites. So we'll talk a little bit more about if they're parasites or not in a bit. And the reason that that happens is that it probably allows the plant to adapt well to the physiology of a particular host. And that's advantageous because those germinating seeds then will only germinate if they are in the presence of the host that's going to be good for them. And they don't have seeds that are germinating without the right conditions. So they have less mortality. And a dependable host probably allowed for the move away from photosynthesis. So if they know that their tree buddy is going to be there all the time, their fungus buddy is going to be there all the time, then they don't really need to be photosynthesizing anymore. Okay, we talked about this kind of already. Oh, I think I just went back here. Just a moment. There we go. So let's look at what some of these things look like. Here are a few of the species that I see pretty commonly up here in Oregon. I don't know what y'all see in California if it's that much different. But this is one of the most common ones that I see, the spotted coral root. And you may be familiar that it has a lot of different forms, so it can be a lot of different colors. Sometimes it doesn't even really have the spots on it. Getting to species level ID on this one can be confounding sometimes. But it associates with Russulaceae. So Russula mushrooms are these big, colorful mushrooms with brittle gills. They're stipes. Stipe is the mushroom word for stem. They break like chalk. And it associates with Russula, Lactarius, and Gymnomyces, which are all within Russulaceae. And so, you know, I talked about that the hosts are pretty specific. And it may not seem that Russulaceae is that specific, but again, within specific small areas, it does seem to have a pretty consistent host within that area. Okay, here's the ghost pipe, Monotropa uniflora. I am a word nerd, so I love to learn what each of the words mean. Monotropa means one turn, and uniflora is single flower. So there's only one flower per stalk, and the turn is they are kind of a pipe shape when they're first coming up, and then as they are pollinated, the flowers turn up. So that's the turn for that one. And this one is also within Russulaceae, like the other one, with different species of Russula and Lactarius, depending on where you are. Where I am coming from in Oregon, the specific host is Russula brevipes, the short stem Russula. It's a super, super common mushroom, and maybe that's why we see so much of this ghost pipe in this area. And this mushroom is edible. Not that many people go searching for it because it's pretty bland, but it is the host for this parasitic fungus that grows on the mushroom and turns it into a more delicious edible, the lobster mushroom. So all of this orange stuff is growing on this white fungus, turning it into a more delicious edible. So if you are seeing these ghost pipes in the summer, then you might be seeing the lobster mushrooms in the same location in the fall. So that's something that might be useful to you if you are a mushroom hunter. And I see Sherman asking, is there a season for these plants? So yeah, I start seeing the coral roots, especially in the spring when I'm out doing morel hunting. They're often up there. And they'll go into the summer mostly, maybe like the beginning of fall. And you could kind of see the like dried husks of some of the plants for even up to a year later, but they get to be looking pretty mummified at that point. Okay, and here is Allotropa virgata, or the candy stick, the one that first piqued my interest about these. You know, it looks like a candy cane, so cool. And there's a very cool story about figuring out the association of this one with the Matsutake mushroom. The Matsutake mushroom, its scientific name is Tricholoma Marillianum. And this is a nice call to community science. The commercial mushroom picker, so not someone who has an academic background or anything, John Getz is the one who is first attributed with learning about this association with this particular mushroom. So John Getz was doing commercial mushroom picking of Matsutake, and primarily in the pine plantations that were planted as part of a dune stabilization program in Oregon. So he did watch the trees develop over time until they started producing mushrooms. And then he started also noticing this plant pop up and was like, that's very interesting. Everywhere that I'm finding Matsutake, I'm seeing this plant. And then he went to California and hunted for Matsutakes there. And he saw that this plant was and in California, the Matsutakes are associating with tan oak rather than pine trees. And he also saw this plant there. So he knew that it wasn't necessarily just something that was associated with the trees, that it actually had to be associated with the fungus. And so now that he knew how to tell where this plant was, and he could see it, remember I said these stalks can last for a year or so. So he can just look and see if this plant is there, and he knows that there's going to be some good Matsutake spot there next year. He doesn't even have to have the mushrooms fruiting. And he kept this to himself for a little while because he wanted to, know where the Matsutake patches were, and no one else wanted to know. He didn't want to tell anyone else about it. But what happened was that the Forest Service one year was going to take the permitting system and make it privatized. They just didn't have they didn't feel like they had the funding to deal with the mushroom permits. And John Getz was very worried that nobody was really going to be watching what the people were doing who were harvesting Matsutakes, because there was a lot of ecosystem damage that was being caused. People would leave a lot of trash. They would disturb the ecosystem in unsustainable ways. There was, since Matsutake sold for so much, there was a lot of infighting and potential violence associated. So it was really important to him that this be regulated by the Forest Service. So at that point, he shared his discovery with the Forest Service because he wanted them to be able to monitor this in a way that seemed like it would work for them on the budget they had. So they were able to zero in on the Matsutake spots better than they would have been able to otherwise. So that's pretty cool. And then later on, someone came and did the DNA and found that it was indeed Tricholoma Marillianum, the Matsutake, that was associated with it, confirmed what he had observed. Here is Sarcodes sanguinea, the snow plant. I think you all probably see this one a lot in California. I don't see it so much in Oregon. This one associates with rhizopogon, which is a ball-like fungus that occurs mostly underground. It's one of the very largest mycoheterotrophic plants. In fact, look how big this one is. It's larger than this guy's beer. I was just searching for pictures of it online and found this guy and had to include him. He was very excited about being included in my presentation. Okay, and going back to the question of, do they give anything in return? Are they just parasites? So in 2000, Bidar Tondo and the group found that there were much higher than normal concentrations of these rhizopogon fungi near Sarcodes plants. So not just the actual fruiting bodies of the fungus, but concentrations of the mycelium of that fungus. So it seems like the plant is doing something to stimulate the mycelium of that fungus to grow. As far as if that actually does something beneficial for the plant, that is still, or does anything beneficial for the fungus, that's still unknown. So it does make the fungus grow more, but for what end? Like if it ends up just giving more food to the mycoheterotroph and not to the fungus, does that really help it? So that is still up in the air. I was talking to a friend who's really into these, and she had mentioned that there are certain bacteria that will colonize plants that are parasitic to them, and they will also stimulate the growth of the plant that they're parasitizing, not necessarily to any benefit to that plant. So, it doesn't necessarily follow that it's beneficial, but it does do something. There has also been some evidence in 2006, they did a study that found that some of the carbon later on from some of the orchids that are able to photosynthesize went back to the fungus. It was a very small amount of carbon. So if that's really like a normal part of its interaction, hard to say, but there is at least some evidence that this give now, get later strategy might work. So the fungus is getting something in return later. And in 2008, there was a follow-up study that they measured the downflow of carbon over time and found that it was greater than the upflow over time. There's a little bit of problem with that study in that they measured the amount of carbon that was just in the roots and the fungus that was attached to the roots. So they don't really have any way of separating out if that carbon was specifically in the fungus or just in the roots. But it does seem like, it was going in the general direction of the fungus. And much is still unknown in this regard. So hopefully, some people can study this and be able to tell us more later. So in that regard, are they parasites? Well, I did talk to Steve Trudell, who's done some of this research, and he said he was pretty comfortable calling them parasites. Parasite is an organism that lives in or on another organism and benefits by deriving nutrients at the host's expense, often doing some harm to the host. So, we're talking about the previous slide. Is that fungus growth actually doing something beneficial for that fungus? And it's also a little bit difficult to actually determine if there's some harm that is happening to the plant from this interaction, the photosynthetic plant. That's difficult to quantify, right? Because you can't just measure all of the carbon that a tree would usually have and then how much is less from that. So it's a very difficult thing to quantify, but it doesn't seem like they're really giving anything back. So yeah, it's probably fine to call them parasites, but again, more still needs to be researched. Okay, and how can we imply this information? So first of all, I don't think that we need to have a reason to apply this information. I think it's just awesome to learn about how things are interconnected. Oftentimes we are finding ourselves well, how can we make money off of this? How is this useful to humans? And, it doesn't always have to be. But if you are a human doing things that you need it to be useful for, you could potentially locate species of fungi, even when they aren't fruiting, such as people who might be studying rhizopogon. Remember I said those are ball-like fungi that fruit underground. So even when they are fruiting, you can't see them oftentimes. So that can be handy. If you are propagating orchids, this is a really good thing to know because especially if there's some sort of rare orchids that you are trying to conserve, you need to know the fungi that are associating with them. And here is such an example. So this is Cypripedium montanum, the mountain lady slipper. And this can live for 15 years without ever producing flowers. And it is underground a lot of that time, just getting nutrition from fungi. And this is a species of concern. And I volunteer with a Mount Hood wilderness organization, and we found one of these things and stopped logging from happening in that area because we found this. And I found a record of someone online that was trying to reintroduce it into some places, and they were having a really hard time with it until somebody else online told them, hey, it associates with the fungus, so that's probably why you're not getting your seeds to germinate. And so they were like, oh, that's great. We're going to try and introduce the fungus next time. Whether or not that can be done is also a little complicated, but it's good to know the ecology of how these things grow if you are trying to put them back into habitat. What fungus are the lady slippers associated with? Chris asks. I do not know offhand. I would have to look that one up. I'm sorry. It's something that doesn't make a fruiting body. So it's just something that happens underground and doesn't look like a mushroom. So it's kind of an obscure genus. And And Sherman asks, are there insects reliant on any of these plants? So I know that at least with the ghost pipe, the bees are pollinating them, so they're doing something. I don't know if there are any that are actually reliant exclusively on them. It doesn't seem like it would be a good technique because they're so uncommon relative to other plants. And with that, I just wanted to tell you all of a couple of resources. So this mycoheterotrophic plants list is on Mushroom Observer. Mushroom Observer is a community science website where people upload their observations of mushrooms to show distribution, etc. And there is a list up there of all of the known mushroom associates of particular mycoheterotrophic plants. So that makes it really kind of easy to figure that out. And I have some references here. I really wanted to thank Steve Trudell for giving me resources. John Getz, who is the one who figured out the whole thing with the Matsutake. He wrote me a very long story about it, which I really appreciate. And Soren Weber's the guy that was going like this with the plant and the beer. And Robin Rietzi has the beautiful photos of the candy stick. And so Susan had asked specifically if I could share some ways to differentiate hemitomes, the gnome plant, the Pidiopus californicus, I'm not sure if that has a common name, and Pleurocospora, the fringed pine sap. And so I did find y'all a key of how to figure that out. The gnome plant has fused petals. So if you look at the very base of the petals, they're fused. And if they are not fused, then it's one of these other two. And one of them, the Pidiopus, is hairy on the inside of the petals. And the Pleurocospora, the fringed pine sap, is not hairy. It is bald. So that's how you tell the three of those apart, which can be very difficult to tell apart. They're all kind of like pinkish, yellowish, and with, circular, fluffy kind of things. It's hard to describe. So I will now take some of your questions, and it looks like there's already a couple here. Gordon asks, can forest stand replacing events such as clear cutting and large-scale wildfires reduce the abundance and distribution of mycoheterotrophic plants? Yeah, definitely. I don't know that there's been specific research about that, but I mean, it definitely stands to reason that that would be true because they're trees, and the associated fungi that are burned in the soil would stop being there. And so the mycoheterotrophic plants would also stop being there. And he also asks, what about shrubs? Do some mycoheterotrophic plants associate only with generally with shrubs? As far as I know, it's only with trees. So they are associating with ectomycorrhizal fungi. So these are ones that generally produce fruiting bodies. So they have mushrooms and they associate with trees. There are some kind of like woody shrubs like Manzanita that can also, maybe Manzanita isn't a good example. Anyway, that can form ectomycorrhizae as well. But none of the ones that I can think of happen to be associated with mycoheterotrophic plants. So trees. Thanks, Griff. Let's see. Okay, if a mycoheterotrophic plant doesn't come up where you saw it before, did the fungal host die? Hard to say. I mean, it maybe, or maybe there was just something about that growing season. Maybe come back to it the next year and see. I don't know. That's a good question. Chris asks, do you know generally how deep in the soil the mycelium from the, I assume the associated fungi is, and I think that it's within a foot or so. If you can, so when I dug down for the ghost pipe roots, it was, I would say, 8 inches-ish down. So I don't know exactly how far they go. It probably just depends on how far the tree roots go that it's associated with. But the connections to them are probably happening fairly close to the top of the soil. Hey, thanks, Brandon. Brandon has a question. In your experience in a forest, how much does the diversity of mycorrhizal fungi differ? What types of places have the highest fungal diversity? So I do have some experience mushroom hunting in different places, and it seems like the mycorrhizal fungi, just the diversity of them, seems bigger in the East Coast, probably because there is a greater diversity of different tree types. So that is my guess about that, but I don't know for sure. And what types of places have the highest fungal diversity? So my guess is that that's probably actually in the tropics, and not necessarily because of mycorrhizae, but because of the diversity of plants that are growing with there. And so a lot of different fungi are associated with different plants, whether or not they're eating the wood or whether they're mycorrhizal or whatnot. Just the diversity of plants and different diversity of mini ecosystems in the forest is going to lead to different fungi in different places. I think I answered Chris's question. Okay, are there any mycoheterotrophic, Jen asks, are there any mycoheterotrophic plants associated with members of Ericaceae? I don't think so, because I think that the fungi are associated with coniferous trees in general. Yep, that's the best answer that I have for that. But I could be wrong. I'm not 100% sure. Jan asks, does collecting the mushrooms damage the other plants? Great question. So the best info that we have about collecting mushrooms is that it doesn't seem to do any damage to the mycelium of the fungus. So it's just like if you're picking an apple off of a tree, you don't do any damage to the tree just by picking the apple off. It's just the fruiting structure. So I would assume that the same goes with the associated plants. That it doesn't really do any damage to them because the mycelium of the fungus is still going strong, whether or not anyone picks the mushroom or the mushroom eventually rots off, etc. CNPS asks, can we call them mycotrophs for short? It's fine with me. I would like that. I think mycoheterotrophs is kind of a mouthful. And Jana had asked about the mushrooms. If so, are there better ways of collecting than others? You might be talking about like the pick or the cut or pull debate. It doesn't make any difference whether or not you cut mushrooms or pull them out. We have some data that suggests that it makes no difference. They've done studies where they pulled or cut or left all of the fungi in any given area, and as far as fungi fruiting in next years for many years after, it doesn't seem to make any difference one way or the other, so do it however you want. Oh, so Jen says except for tan oaks. Yeah, so members of Ericaceae that are associating. Is tan oak in Ericaceae? Y'all are plant people. I see some shaking heads. Okay. Oh, because I said it mostly associated with conifers. Right. Yes, thank you for that. Great, thank you, Annie. Robert asked, what are some of the fungal species are generalists serving many plants? What are some of the fungal species are generalists serving many plants? So there are definitely ectomycorrhizal fungi that can associate with more than one kind of plant. So they are not as specialized. The russulas, for instance, can associate with different conifers. One individual fungus can potentially be attached to trees of different sorts, whether they be broadleaf trees or conifers. So it is true that they are not as specialized. Some of them are, but not all of them. Okay. Bill says, fascinating talk, reminds me of the work of Susan Simard. Yeah, and if people want to know more about her work, and she has dubbed the wood wide web, where trees actually communicate with each other through this fungal network that they're attached through. Highly recommend looking into that more. There is a radio lab episode that I think is called From Tree to Shining Tree that I would highly recommend that talks about that. Brandon says, out east where I am, many mycoheterotrophs associate with deciduous trees like beech, maple, and oak. But it does seem like out west it's more on mycorrhizae associated with conifers. Yep, so I guess I'm talking specifically about the mushrooms that I know around here that associate with them. And in Oregon, there's a lot more conifers than there is out east, just in general. In the Pacific Northwest, very conifer heavy environment. So yeah, it's going to differ depending on where you are. Okay, I think that's all the questions so far. Any more? Okay, Lisa asks, are there any of these similar plant fungal relationships in the drier areas of Southern California? I guess I don't know that because I haven't hunted for any of these things there, but maybe somebody else in the chat can say if they have seen any of these mycoheterotrophic plants there. Kevin asks, is there always a large distance between the carbon donor and the mycoheterotrophic plant? Could the donor and mycoheterotrophic plant be in the same genus? Yes, there's a large distance because it's usually a tree and then, some member of Ericaceae. So I don't think, as far as I know, no, they definitely couldn't be in the same genus. Because then you would have another mycoheterotrophic plant, and that the two of them would not be able to share any food because they're stealing food from everyone else. So no. Stop my share here.
Well, Leah, that was just great. I think you can tell from the questions that you really got people thinking.
Yeah.
It's thinking about a part of the natural world that we just don't see. We have to go by these little clues. When a mushroom pops up here or a mycoheterotroph pops up there, they're telling us that something's happening out of sight under our feet.
Yeah, definitely. And still so much to be known. And Brandon says there are no fully mycoheterotrophic species in deserts out west. There are root parasites on other plants though, but Monotropa does live in dry parts of Florida scrubs. Okay, so it must be associating with some sort of little like shrubby thing or pine trees. Great.
Okay, now everybody will be out there next year looking for mycoheterotrophs. And probably looking for mushrooms before that, right now.
Right. Well, and you can, right now you can mark those spots if you see the candy stick, the old like dried up ones, and you might be able to find matsutake there later. Right now, our matsutake is just starting to pop up where I live, and I was in an area the other day where you can see all of the dried out husks of the candy stick there.
Yeah, I use those dry plants a lot. I think I can tell apart the different species of coral root based on the old petals that are still attached to the empty fruits on the dry
Wow.
stalk.
That's impressive.
The flower parts are there. You just have to look.
Yeah. Wow. Awesome. Terry, you do have your hand up? Yeah.
I do. I just put it up because I remembered something that I hadn't remembered earlier in your talk. I wanted to ask you a question based on this memory. There was a paper about 15 years ago or so looking at possible mycorrhizal associations with this group of neotropical plants that are in the Gentianaceae.
Mm-hmm.
The genus Voirea is one. I see them a lot in my field sites in Guyana. Super reduced aerial plant body on the Voirea, one flower at the tip of about a 2-inch tall stalk.
Mm-hmm.
They're usually brightly colored, but they're definitely achlorophyllous. They're not green.
Okay.
And I think Tom Bruns and some of the folks at Berkeley were involved in this study. They figured out that it is associated, the Voireas are associated with a mycorrhizal fungus, but it's a glomalien mycorrhizal fungus, like an endomycorrhizal fungus. And I don't think they were ever able to determine if it was actually connected to a photosynthetic plant nearby. But they're achlorophyllous and they have glomalien or endomycorrhizal associations, putatively connected via the AM fungus with some photosynthetic plant nearby.
Mm-hmm.
Have you heard of any instances of that occurring in the temperate zone? Like an mycoheterotrophic plant associated with a glomeromycete fungus?
Yes. So I believe the orchids that are not fully mycoheterotrophic, that is often the case.
Cool.
Not just here, but elsewhere as well.
Hey, could I jump in? This is Brandon.
Yeah.
Yeah, Terry, the Gentianaceae, just like how orchids associate with fungi, the gentians also have very small seeds, even the green ones. And there's partially mycoheterotrophic gentians all over the East Coast that associate with arbuscular mycorrhizal fungi. So one of them is called Bartonia.
Okay.
And it looks just like Voirea, and we have Voirea in Florida as well, so subtropics. Just a green little stem with little nubby leaves. So I think that one is associated with AM fungi. With the AM fungi, it's mostly things in the tropics. There was another interesting case. So Thismia, which is kind of a southern hemisphere and tropical genus or a family, excuse me. There was one that was found in Chicago in the early 20th century, and it's thought to have gone extinct, but that family is also associated only with AM fungi. So it was in like an oak savanna, probably with AM fungi and only known from Chicago. So that's one of the weirder stories out there for those kind of plants.
So Brandon, do you know if anybody's ever demonstrated the interconnectivity in these associations, like definitively shown that what other photosynthetic plants they were actually connected to?
Yeah, the difficulty with all of this, and Leah did a really great job of kind of showing the historical studies on it. It's just that it's so difficult to grow the tripartite relationship in a greenhouse or something. And there's been times when they've tried to culture the seeds in a permeable bag and see it get colonized, but but it's very difficult. So it's mostly done through indirect measurements of the isotopes of a tree and of a
Yeah.
plant. Similar, that's what I do. But I think that the assumption is that it is connected to a tree. So a lot of them are associated with these trees in the family Diptocarpaceae, which is like a Southeast Asian. So those same glommalian fungi are associated with those trees, and also a lot of mycoheterotrophs in those soils, in those, in Borneo and Thailand. So yeah, I think that's what the assumption is. Whether they've actually seen it connected, I'm not 100% sure about that.
Cool. Yeah, well, I just learned something, that they're more widespread, these glommalian mycoheterotroph associations, than I had thought. Nice.
Yeah, so with something like that, maybe they would take the root and then, do the DNA to find what all of the different fungi are that are inside of the root, right?
Yeah, and that's partly what I do. But you end up getting hundreds and hundreds of fungi in the roots, and I'm sure you've seen this. And when you read these papers too, right, Leah? Like they'll give a whole list of what they found inside of the roots. So what we know about roots of plants and the microbiome, it's really evolving. And we haven't even started to talk about bacteria in the roots either, and what are the bacteria doing. So it's kind of an open question. But yeah, if you look at all the mycoheterotrophs, most of them are orchids, and the orchids, as Leah was mentioning, mostly associate with the ectomycorrhizal, endomycorrhizal fungi. But if you look at the other families apart from the orchids, most of them associate with arbuscular mycorrhizal fungi. So it might be something to do with just there being more arbuscular mycorrhizal fungi associated trees in the tropics. And then in the temperate areas, it's more conifers, and those are mostly associated with endo and ectomycorrhizal fungi, right? So it might be something to do just with the forests trends in general.
Yeah.
But yeah, I think that all this stuff is super fascinating, right?
Thank you so much for chiming in there. It's nice to hear from someone who's doing the research.
I was very excited to see this talk, and I think you did a really great job summarizing it. So yeah.
Thank you.
Thank you for that.
Yeah, I'll agree that as the novice coming in, it was a real good beginner's lecture, complete with up-to-date information and telling how the experiments were done. That's just great.
Thank you.
Maybe it's going to inspire somebody to go out and do some experiments themselves.
I hope so. Get back to us about what these things are doing.
Okay, well, I guess we're saying thank you.
Thank you, Leah.
Thank you.
It was a great evening, Leah.
Indeed. Thanks so much.
Thank you for having me.